Hi, I am Alicia Mastreta Llanes from Conavio, Mexico. Today I'm speaking on behalf of the Coalition of Conservation Genetics and in coordination with a large group of conservation biologists and geneticists. Today I want to present in this video a short summary of a recent research showing that the genetic diversity indicators are developed, tested, and ready for implementation. The Global Biodiversity Framework aims to conserve genetic diversity, not only of domesticated species and their wild relatives, as it was done before, but for all species, including wild, domesticated, and not necessarily of economic importance. Genetic diversity is included in Goal A and Target 4. I'm not going to read you the thing, you can check it here. But I want to highlight that we aim to conserve and restore genetic diversity within and between populations. And for that, we need indicators to monitor genetic diversity. I know that this sounds scary, but the genetic diversity indicators that were adopted during COP15 are simple. Data is already available, and importantly, we don't need genetic data to estimate them. To illustrate this, we undertook a first assessment in 9 of our home countries, which are very different in terms of biodiversity and the type of research infrastructure. And we were able to estimate the indicators in more than 900 species with 5,000 populations within them of very different taxonomic groups, including insects, mammals, plants, and so on. Data was available for the majority of the species. I will tell you more about that later, but first let's see some of the results. One of the indicators refers to the proportion of populations maintained and was adopted as complementary indicator. This refers to the loss of genetically distinct populations because when a population goes extinct, we lose whatever genetic diversity was within it. And we don't need genetic data to know this. We just need to count how many populations were there, for instance, fishes in different lakes, and how many remain. In this indicator, we found that 40% of species have lost at least 1 out of every 10 of their populations. Fortunately, the other 50-something% of species maintain all their populations. This is good news. However, let's see what happens with the other indicator. The other indicator refers to the proportion of populations within a species with an effective population size above 500. Why 500? This is a number, a threshold, where populations are known to be too small to retain genetic diversity. And this means that diversity will be lost even if populations remain. This was adopted as the headline indicator. The effective population size can be estimated with genetic data, but if we don't have genetic data, we can also approximate it based on the number of mature individuals and a ratio. For instance, for many species, this ratio is around 10%. So 30 mature frogs equals an effective population size of 3. In this indicator, we found that in 58% of species, all populations are too small to sustain genetic diversity, meaning an indicator value of 0. And this is very serious because it means that populations are losing the genetic diversity that they need to adapt to changing conditions like climate change and pollution. I want to highlight that we were able to estimate these indicators using data already available in published studies, our local databases, citizen science, and asking local people about the populations they know. The indicators do not need genetic data. The effective population size can be estimated based on the number of mature individuals, and populations can be defined based on geographic features or biological features. Actually, in our study, the majority of the data to estimate the effective population size came from point estimates or range estimates on the number of mature individuals and then applying the ratio. And this is perfectly fine. Only around 6% of the species we analyzed have data to estimate the effective population size based on DNA data, but this doesn't matter. We could estimate it based on the other ratio. So I hope you enjoyed this video. If you want to know more about our results and our methods, please visit our website. We have a special section for SABSTA with a link to a preprint and the policy brief of our results. Thank you so much for watching.